there were two things. When I first went to USC, we spent a lot of time trying to understand how class 2 functioned in antigen presentation. There are still sessions going on at this meeting about how it works in antigen presentation, how you put peptides on, and trying to understand both the primary kinds of professional APCs and ones that went on in other tissues. And what drove me is we started a collaboration with Lee Hood to clone these genes. And what we started to do at USC, and I continued at the beginning when I moved to UNC, was to do detailed structure function studies on MHC class 1 molecules by doing mutagenesis. And we actually did those in mouse for a long time. And when I went on sabbatical to Oxford, I moved into doing those in humans. So we did them in mouse and we did them in human and in Oxford. And that was really occupied the lab probably for 15 years, trying to drive that problem into the ground. We wound up, because in humans, a really good way to look at that was to study HIV. So we spent a lot of effort studying HIV in terms of how CTLs worked in HIV to control virus infection, how MHC polymorphisms dealt with that, and how to think about trying to make better potential vaccines. I think one of the interesting things, and one of my favorite stories that I like to tell Congress critters, is as we were doing this, we were working with Ed Collins, the crystallographer, aforementioned crystallographer, and we were trying to figure out really the biophysics of how peptides stuck onto MHC. And Ed came across this breast cancer epitope that's in HER2, and you may have heard of Herceptin, which is, right, and it's one of the targets of Herceptin. But it's also a target for cytotoxic T cells. And Ed and I were going to do a structure, and it was just a terrible epitope. It didn't bind very well. You had to do it at low temperature. We said, well, this sucks. We just got to fix this. And so we realized that from the HIV stuff that we had done, that we could make stuff that was better than natural. That is, we could make peptides that stuck better. That is, we changed. So peptides bind like a hot dog in a hot dog bun. And so you can change the things that stick down, you can change the things that stick up. So for the T cells to work, you got to leave the things that stick up the same, but you can make the stuff that sticks down gluier. So we did. And together with John Soroti, who had also was a hematologist oncologist, who'd also been a fellow in my lab, we devised a clinical trial. So we published a bunch of papers where we showed that, yeah, you can do this to HER2 and it's better and it's more immunogenic and all the things you want to be. And John organized a real dendritic cell therapy altered peptide ligand trial to do this. And it was sort of my first real human clinical trial thing. And of course, the very first patient we treated had an objective response, which means her tumors got smaller. And the next 13 did not. So in terms of changing clinical medicine, not so much, but in terms of taking a problem from biophysics, right, to a patient, it was really good. And that was something which I could really only do in a place where I had lots of connections, where there was a lot of interest in collaborating across disciplines and a lot of interest in being able to drive things from ideas to medicine.
And that was at USC?
That was in UNC. It was in Chapel Hill. Right. And so that's kind of what we did. We did a lot of other stuff too, as my unfortunately varied CV will show. But I think this is the kind of thing which I really liked about UNC.
Well, you were there until just
2 years ago.
Right.
Yeah, almost exactly 2 years ago.
Right. So there must have been other breakthroughs, no?
Yeah, no. So one of the things that we did with Roland Tisch is, one of the questions in autoimmune diabetes is where does the immunology happen, right? And so the conventional idea is that all immunology happens in lymph nodes, and the effector cells leave lymph nodes and go to the target tissue. In the case of type 1 diabetes, those are the islets of Langerhans and the pancreas. What we said was, okay, well, what we're going to do is we're going to look by sequencing the T cell receptors in the pancreatic lymph nodes and in the islets and ask whether they're the same. So if all the immunology goes on in the lymph nodes, those should be essentially the same distributions, right? You should have the same mix of kinds of cells. When we did that, what we found is, well, there were some that were the same. There were a whole bunch that were only found in the pancreas, were not found in the draining lymph node. So if you're going to save conventional wisdom, you have to wave your hands really fast, and you have to say, oh, they used to be there, right? But they left. Okay, so we're still doing experiments to drive that, but what we really did show is that almost certainly there are cells that are being developed in the pancreas independent of the pancreatic lymph node. And the specificity of those is really important if what you want to do is block the progression of type 1 diabetes. So during the disease course, for walking around human beings, people become glucose intolerant. And then if they control their diet, there's some rebound of their ability to control blood sugar. And that, the clinicians call that the honeymoon period. And then eventually enough more beta cells in the pancreas get destroyed and you can't make enough insulin to regulate. So the idea, if we could block progression at that point of disease, that would be really good, because we could clearly regulate blood glucose better. We could clearly have people not take so much insulin. We could clearly fix a lot of the downstream sequelae of type 1 diabetes, which are really bad, right? So diabetics is the leading cause of amputation, right? You get retinopathy, so you go blind. People view these as bad side effects, and we call those poor prognostic indicators, right? And so what we wanted to try and do is to be able to look at those, and we're still doing those in Arizona, those kind of experiments in Arizona, both in mice and trying to be able to get samples to do it in people, to ask that same question about the diversity of the T cell repertoire. We did lots of other amusing things. We had a really nice collaboration with Lishan Su, who is one of my colleagues in the Cancer Center, looking at what people now call BLT mice, bone marrow, liver, thymus reconstituted, Camasi null SCID NOD mice reconstituted with human fetal liver and human thymus and human bone marrow. And those mice generate a pretty good human immune system, but they also have human liver. And what Lishan did was, and we published a paper last year that came out in Gastroenterology showing we could infect those mice with hepatitis B, and we could get a human immune response in those mice. So that now gives you a way to study pathogenesis of hepatitis C without poking needles into walking around people. And it gives you ways to test therapies that you can't do otherwise. So we're pretty excited about those kinds of things, and we published a paper with Roland and some of the clinicians at UNC doing a similar thing in type 1 diabetes. So we're really interested in trying to develop these kinds of models where you can mimic a human disease with a human immune system in an animal model that you can get access to the important parts.